7 states in America have just made it a crime to sell this: Florida, Texas, Alabama, Mississippi, Nebraska, Montana, Indiana. And yet, the federal government is actively pushing to put it on every grocery shelf across this country. Today, I'm going to walk you through exactly what this new technology does inside your body, and specifically what it does to your stem cells, and explain why you're hearing so little about the real science behind it. I'm Dr. Becker. I've spent years in clinical practice watching how what we eat shapes what happens inside ourselves, particularly in people over 50. What I'm about to share comes from peer-reviewed research from published studies in journals like Frontiers in Nutrition, from researchers at MIT, and from the legislative record of 7 state governments that looked at this evidence and decided the safety case had not been made. As always, what I share here is educational. If you're on medication or managing a diagnosed condition, please talk to your doctor before making any significant dietary changes. That's not a disclaimer I say to protect myself. It's something I genuinely mean. Individual health context matters, and no video, however thorough, replaces a conversation with someone who knows your specific history. This is not a political video. I genuinely don't care which party you vote for. What I care about is what the science actually says. And right now, the science raises questions that nobody in the mainstream food conversation wants to answer honestly. By the end of this, you'll understand what cultivated meat actually is at the cellular level, what the published research says about the genetic processes involved in producing it at scale, why your age, specifically being over 50, matters enormously in this particular conversation, and what you can eat instead to actively support the stem cells you already have. Let's get into it. There is a question at the heart of this entire debate that the industry has never publicly answered. What happens to a human body that regularly consumes cells engineered to divide indefinitely? Not for 3 months, not across a short-term regulatory review, but over 10 or 20 years of consistent daily consumption. The honest answer from the scientists themselves who have studied this most carefully is that nobody knows. Nobody has done that study. Nobody has followed the population for 2 decades and measured the outcomes. And the federal government cleared this product for your dinner plate before that answer exists. That fact alone is worth sitting with before we go any further. If you're finding this kind of investigation valuable, a subscription here helps me keep doing this work. No pressure at all. I just want you to know it makes a real difference. Let me take you to the beginning of the science, because to understand what the concern actually is, you have to understand what cultivated meat really is at a biological level. This is not a plant-based product. It is not texture soy protein or a microprotein derived from fungi. Cultivated meat, also called lab-grown meat or cell culture meat, is produced by extracting living cells from an actual animal, typically muscle satellite cells or adipose stem cells, and placing them inside a large industrial container called a bioreactor. In that bioreactor, those cells are based in a liquid nutrient medium containing amino acids, glucose, vitamins, minerals, and critically growth factors. They are stimulated to divide, multiply, and eventually form tissue that structurally resembles meat. The entire commercial goal is to scale this process to produce enough product cheaply enough to compete on supermarket shelves with conventional meat at mass market prices. On paper, it sounds like a clean, efficient, even elegant solution to some genuinely difficult problems. No slaughterhouse required. Dramatically reduced land use. Protein at industrial scale with a fraction of the carbon footprint of conventional livestock agriculture. I understand why people find it appealing. I also understand why investors have poured billions of dollars into it. But the problem is not the goal. The problem is what has to happen to the cells to make them divide at the speed and volume that industrial production requires, and what that process does to the genetic identity of those cells in the process. Here's the biology that never appears in the press releases or the investor decks. When you take normal muscle cells from a chicken or a cow and place them in a laboratory environment, they follow natural biological rules. They divide a limited number of times. There is a ceiling built into every healthy cell, a limit on how many times a given cell can copy itself before it stops dividing. This ceiling is called the Halleck limit, named after the researcher who identified it, and it exists for reasons that are fundamentally protective. Cells that divide indefinitely, without a stopping point, are almost by definition pathological. Every healthy cell in every healthy body operates within this constraint. The moment a population of cells loses that constraint and begins replicating without stopping, we have a clinical term for the process that describes it. We call it malignant transformation. In plain language, that is how cancer begins. To produce cultivated meat at commercial scale, companies need cells that ignore the Halleck limit. They need cells that will keep dividing day after day, week after week, filling bioreactors and producing volume continuously. This means the cells must be modified either through deliberate genetic engineering or through extended laboratory culturing to become what scientists call immortalized cell lines. Lines of cells that do not stop dividing. The terminology itself should prompt careful thought about what is actually being placed in the food supply. A 2024 paper published in Frontiers in Nutrition by researchers Hauser, Searman Rack, and Nachman examined this challenge directly and thoroughly. The title was Challenges and Opportunities in Cell Expansion for Cultivated Meat. What they found and published in a peer-reviewed journal is that to achieve the continuous division required for industrial production, the cells need their natural biological brakes on growth to be overcome. The approaches described in the paper include altering genes that regulate the cell division cycle, specifically like TP53 and P10. I need you to understand what those genes actually do, because they are central to everything that follows in this conversation, and they represent the biological core of the concern. TP53 is the most studied tumor suppressor gene in all of oncology. It is sometimes described as the guardian of the genome, and that description is entirely accurate. When your DNA sustains damage from radiation, from environmental toxins, from errors that naturally accumulate during routine cell division, TP53 is the molecular mechanism that detects that damage. Once it detects the damage, it does one of two things. It pauses the cell cycle to allow DNA repair machinery to correct the error, or it triggers programmed cell death apoptosis to prevent a damaged cell from replicating and passing its errors forward into daughter cells. TP53 is one of your body's most fundamental biological protections against the development of malignancy. Mutations that inactivate TP53 function are found in more than 50% of all human cancers across all tissue types. It is not an obscure regulatory gene. It is the single most clinically significant tumor suppressor that cancer biology has identified in 6 decades of research. P10 is the second gene named in the paper. It is another tumor suppressor that regulates cell proliferation and survival. And like TP53, it is among the most commonly mutated genes across the full spectrum of human malignancies. When P10 function is lost or reduced, cells receive a persistent pro-growth signal that drives them toward uncontrolled division, particularly through activation of the P3K signaling cascade. The Frontiers in Nutrition paper states this directly and without ambiguity. Quoting from the text, one approach involves altering genes perturbed in cancer such as TP53-P10. Such alteration can be achieved spontaneously by prolonged culturing. That last phrase is the one that should command your attention. Spontaneously. You don't have to deliberately engineer these mutations into the cells. Leave cells in culture long enough under the conditions required for industrial production, and they start accumulating these mutations on their own. The production environment itself creates selective pressure that favors cells that have already partially lost the genetic constraints on a limited division. A separate 2023 paper in the same journal examining genetic drift and stem cell adherence specifically in laboratory culture conditions added a finding that should have generated significant regulatory attention. Researchers found that normal cells from non-primate animals, the type most commonly used in cultivated meat production, develop what the paper describes as a tumoric profile after only 2 passages in a laboratory environment. 2 rounds of cell division in culture. These were cells that began as normal, healthy, functional tissue. Within 2 generations of laboratory culturing, they were already showing genetic characteristics consistent with tumor cell behavior, including evidence of metastatic potential. The paper's specific recommendation was that mandatory genetic quality control testing be implemented for all cells used in cultivated meat manufacturing, with particular emphasis on detecting oncogene activation mutations in the cell lines. That recommendation has not been adopted as a regulatory requirement by the FDA or any other major food regulatory authority. I want to be precise here because intellectual honesty requires precision when the stakes are this high. The cultivated meat industry will tell you, and they are making a technically defensible argument as far as it goes, that consuming cells with altered tumor suppressor genes is not equivalent to eating cancer. Your digestive system is not passive. Gastric acid actively denatures proteins. Digestive enzymes break DNA into small fragments. The assumption underlying the industry's safety position is that none of the genetic material from these cells survives digestion with sufficient structural integrity to exert meaningful biological effects in the consumer's body. That is their stated position, and I'm not dismissing it reflexively. But here is the question that has remained unanswered since this technology entered the commercial market. That safety assumption rests on what evidence precisely? What long-term human consumption studies supported? What population has been followed for 20 years consuming this product and measured for outcomes? The researchers at MIT examined this gap and published their conclusion. In a 2021 paper widely cited in both regulatory and scientific contexts, MIT researchers stated clearly confirming that future products made from immortalized animal cells expressing spontaneous immortalization or genetic engineering would be safe represents a gap in knowledge in this field. A gap in knowledge acknowledged in published research by MIT scientists in a food product, the FDA then cleared for sale in 2023. The FDA's clearance of Upside Foods and Good Meat in 2023 was based on what the agency calls a cell culture consultation process, a structured review of safety documentation submitted by the companies themselves. It did not involve independent long-term randomized controlled trials of human consumption. It did not involve 10-year follow-up data on any defined population. Singapore had approved cultivated meat for commercial sale in 2020, making it the earliest adopter by several years. The longest any commercial population has been consuming these products regularly is under 5 years. The epidemiological monitoring infrastructure is not in place to detect anything beyond acute effects in a consumer base that's small and that recently exposed. The analogy I keep returning to because I think it captures the situation with genuine accuracy. Imagine you are about to board a commercial flight. You ask the engineers whether the aircraft is safe to fly. They tell you it passed all the pre-departure inspection protocols. They have reviewed the submitted technical documentation thoroughly. They are confident based on their understanding of aerodynamic principles that it should fly without incident. But they also tell you that this specific aircraft design has never been tested for more than 3 years of continuous commercial operation and that some of its core components were specifically engineered to override the usual mechanical safety limits that prevent catastrophic structural failure, and that they cannot fully disclose how those components were modified because the modification is considered proprietary trade secret. Would you board that plane? The federal government not only bought the tickets, but is actively promoting the flight to the traveling public. Let me move now to what I consider the most clinically significant layer of this entire story. The layer that is most directly relevant to you if you are in your 50s, 60s, or beyond. And the reason I think the age-specific framing of this conversation matters so much. The concern here is not identical across all age groups. The risk profile is genuinely age-stratified, and the biological reasons for that stratification are not complicated once you understand them. Your stem cells right now carry what you could describe as a biological history, an accumulated record of every round of cell division they have performed across your lifetime. Every time a stem cell divides to repair tissue damage, regenerate organ function, or respond to physiological demands, there is a small but real probability of a replication error, a mutation being introduced and not corrected before it becomes permanent. Your DNA repair machinery catches the vast majority of these, but some accumulate over time. This is not disease. This is not pathology. This is normal biological aging operating exactly as expected at the cellular level. The older you are, the higher your baseline mutational load in your stem cell populations, and the less efficient your DNA damage response pathways operate. The efficiency of TP53 function itself, that guardian of the genome, decreases measurably with age in human tissues. This is one of the primary biological reasons why cancer incidence curves upward so sharply in the 6th, 7th, and 8th decades of life. The architecture that normally detects and eliminates precancerous cells becomes progressively less reliable over time. Now consider with that context clearly in mind what it means to regularly consume a food product produced through processes that involve the deliberate or spontaneous alteration of TP53 and other tumor suppressors in a body where your own TP53 system is already operating with diminished efficiency as a consequence of normal aging. The specific scientific question of whether that combination has any meaningful biological effect on the consumer has simply not been asked. No study design has examined it. No population has been followed. The absence of research on a question is not evidence that the answer is favorable. It is evidence that nobody has looked. And the population with the greatest concern, people over 50 who are making careful health-conscious dietary decisions precisely because they understand that their biology needs more support than it did at 30, is exactly the group for whom this evidentiary gap should be taken most seriously. The second biological mechanism that deserves careful attention here is growth factor signaling. And this one connects to something you may already be familiar with from longevity research. To keep cells viable and continuously proliferating in bioreactors across the production timelines required for commercial scale, cultivated mid-manufacturing requires continuous delivery of growth factors, specifically insulin-like growth factor 1, which researchers abbreviate as IGF-1 along with fibroblast growth factor and epidermal growth factor. These are molecular signals that tell cells to grow, to divide, and to survive. In normal human physiology, they are tightly and precisely regulated. Your body produces them in response to specific physiological triggers, exercise, tissue repair, growth states, and then downregulates them when the triggering need has resolved. The pulsatile context-dependent nature of that signaling is physiologically important. In industrial cell culture for commercial meat production, these growth factors are present continuously and at concentrations required to sustain indefinite proliferation. The question that bears directly on consumption is whether residual growth factor activity persists in the final cultivated product and whether regular ingestion of cells that have been continuously saturated with procroproliferative signals has any measurable downstream effect on the growth factor environment of the person consuming them over months and years. The reason this matters with particular force for people over 50 is that the relationship between circulating IGF-1 levels and cancer risk in older adults, one of the most thoroughly documented associations in all of cancer epidemiology. The data is consistent across multiple large cohort studies and has been the subject of major meta-analyses published in high-impact journals including Lancet Oncology. Elevated IGF-1 is consistently associated with significantly increased risk of prostate cancer, breast cancer, and colorectal cancer, with the strength of the association increasing in people over the age of 50. The mechanism is well characterized. IGF-1 activates the P3K-ACT-mTOR intracellular signaling pathway, which promotes cell survival and actively suppresses the apoptotic processes that normally eliminate cells showing early premalignant changes in a body, where tumor suppressor function is already reduced by age-related changes. Persistent or elevated activation of this pro-survival pathway in the wrong cellular context is precisely the mechanistic sequence through which malignant transformation progresses. I want to say as clearly as I possibly can that I am not claiming cultivated meat causes cancer. The published data does not support that statement, and I will not make claims the data does not support. What I am stating carefully based on what is actually in the literature is that the specific biological processes required to produce this food at commercial scale involve the genetic and molecular pathways that 60 years of cancer research have identified as centrally relevant to malignant transformation in aging human biology. And there is no published long-term safety data on human populations consuming this product across years or decades of regular exposure. Both of those statements are independently and verifiably true. Together, they describe a situation that warrants caution, not dismissal, not panic, but serious sustained caution rather than regulatory approval based on document review and a safety assumption that nobody has actually tested. The third mechanism I want to address is consistently underemphasized in discussions of cultivated meat. Perhaps because it's less dramatic than genetics and growth factors, but is in some ways more immediately relevant to how you feel and function day to day. Real animal food, specifically grass-fed pasture-raised meat, is not simply protein in a structural matrix. It is a biologically complex substrate that carries a native microbial community of bacteria and archaea that have co-evolved with mammalian digestive biology across evolutionary time. It contains active enzymes that support the digestive process. It contains connective tissue rich in glycine, proline and hydroxyproline amino acids that serve not only structural functions, but act as signaling compounds for minimal stem cells involved in maintaining the integrity of joints, bones, tendons, and the gut lining. The fat in grass-fed beef contains conjugated linoleic acid, CLA, at concentrations substantially higher than grain-fed beef. And CLA has demonstrated activity on tumor suppressor pathway activation and inflammatory resolution in multiple studies published in peer-reviewed nutrition journals, including the American Journal of Clinical Nutrition. The fat also carries meaningful concentrations of vitamin K2 in the MK4 form, a compound that plays a specific and documented role in directing calcium into bone tissue and away from arterial walls. A distinction with direct clinical significance as vascular calcification risk increases after 50. Cultivated meat grown in a sterile bioreactor under laboratory conditions is biologically impoverished by comparison to the food it's designed to replace. It is muscle fiber architecture without an ecosystem. It is protein without the biological intelligence that real food has carried into the human diet for thousands of generations. For someone over 50, where gut microbiome diversity has already declined from its peak, where digestive enzyme production has already decreased, where the efficiency of fat-soluble nutrient absorption is already reduced, replacing a food that your biology has a 10-all-on-year relationship with, and substituting something that has existed for under a decade without long-term safety characterization is not a nutritionally neutral act. The fourth concern entered the public record explicitly through the Texas legislative process, and it connects to one of the most rapidly growing areas of environmental health research: microplastics. Industrial bioreactors are manufactured from plastic, The cells that become cultivated meat are grown direct, sustained continuous contact with plastic surfaces, plastic-lined vessel walls, and in many production systems, plastic-based scaffolding materials that provide the structural framework on which muscle cells organize into tissue. Members of the Texas House of Representatives specifically cited as part of the documented rationale supporting their prohibition legislation that the production process for cultivated meat involves direct interaction between cellular matter and microplastic materials with rupture for tension of cellular membrane integrity. This is not an obscure or fringe scientific concern in the current research environment. Over the past 3 years, the peer-reviewed literature on microplastic biology has expanded dramatically. Microplastic particles and associated chemical compounds have now been detected in human blood samples, in pulmonary tissue biopsies, in arterial plaque specimens, and in human placental tissue. The interaction between microplastic particles and mammalian cell membrane function is an active and rapidly expanding area of investigation. And here is a food production system that places the actual cellular material that becomes the food in continuous contact with plastic surfaces at elevated temperatures under growth factor stimulation for extended production periods. The characterization of microplastic incorporation in the final cultivated meat product has not been published in the peer-reviewed literature. Let me address the financial architecture of this industry, because understanding who is making money from this technology and how they have structured their legal protection of it is directly relevant to understanding why the regulatory pathway moved as quickly as it did and why certain questions remain unanswered. Bill Gates is the largest individual investor in cultivated meat companies. That is a documented matter of public record, not a rhetorical talking point. Tyson Foods, the dominant conventional meat processor in the United States, has invested directly in Upside Foods, which is the company whose cultivated chicken product received FDA clearance in 2023. JBS, the largest beef processing company in the world, acquired a cultivated meat startup as part of its portfolio diversification. These are not idealistic entrepreneurs operating outside the existing food production establishment and disrupting it from below. These are the largest incumbents in conventional food production globally, positioning their capital to control whatever system replaces the one they currently dominate. Their financial interests are structurally aligned with rapid regulatory clearance and commercial deployment, not with waiting 20 years for longitudinal safety studies. And then there is the intellectual property issue, which I find generally alarming when you understand its implications. Several of the leading companies in this space have made a deliberate and considered decision not to patent their core production processes. Patents require full public disclosure. When you file a patent, the entire methodology, every genetic modification to the cell lines, every characteristic of the growth medium, every chemical compound involved in the production process becomes part of the permanent public scientific record. Regulators can review it. Independent researchers can examine it. Journalists can report on it. Consumers can read it. By choosing to protect their production processes as trade secrets rather than filing patents, these companies have ensured that the public will permanently lack access to the specific details of how the cells in the food they are eating were modified and maintained. The House paper in Frontiers in Nutrition from 2024 documented this pattern explicitly. Companies are deliberately structuring their intellectual property strategy to prevent the disclosure that patent filing would otherwise require. In any other domain of food safety regulation, a situation in which the production methodology of a novel food is permanently withheld from public and regulatory scrutiny would be considered a serious and unacceptable transparency failure. In cultivated meat, it has been treated as routine commercial practice. Ron DeSantis, when he signed Florida's prohibition into law in 2023, used language about global elites and the plan to force populations to eat insects and laboratory products that made it straightforward for critics to categorize the entire legislative act as populist theater rather than substantive policy. I understand why that framing was deployed and why it landed, but strip away the political rhetoric entirely and examine what the act of 7 independent state legislatures actually represents at its core. 7 separate governmental bodies, each conducting their own review of the available scientific evidence, each independently reaching the conclusion that the safety case for commercial authorization of cultivated meat had not been sufficiently established to justify allowing its sale to the public in their jurisdiction. Whatever your assessment of the political figures who signed those bills, the underlying regulatory determination is documented, substantive, and grounded in the same evidentiary gap, the absence of long-term safety data that the MIT researchers acknowledged in their published work. 7 states decided that absence was sufficient reason to wait before authorizing commercial sale. The federal government decided it was not. Now, let me focus on what you can actually do with this information, because the practical question always matters more than the theoretical one when you are making real decisions about what to eat every day. The biology that supports healthy stem cell function responds to dietary inputs that carry thousands of years of human use and an expanding evidence base in the contemporary research literature. For people over 50 specifically, here is what the published science supports. Grass-fed beef from cattle raised and finished entirely on pasture from birth through harvest provides a nutritional profile meaningfully different from the grain-fed beef that dominates conventional retail. The CLA content conjugated linoleic acid is measured in concentrations 2 to 5 times higher in grass-fed than grain-fed beef across multiple comparative analyses varying by season and regional forage quality. CLA has demonstrated activity on tumor suppressor pathway activation and prostaglandin-mediated inflammatory resolution in peer-reviewed research including multiple papers in the American Journal of Clinical Nutrition. The vitamin K2 content, specifically in the MK4 form that is most biologically active in humans, is present in meaningful concentrations in the fat of grass-fed cattle and is largely absent in grain-fed beef fat. MK4 plays a specific and documented role in carboxylating osteocalin and matrix GLA protein reactions that direct calcium toward bone mineralization and away from arterial wall deposits. This distinction has direct clinical relevance as vascular calcification risk increases progressively after 50. Practical guidance for implementation: 85 to 140 grams per serving 3 to 4 times weekly. Do not trim the fat. A disproportionate share of the unique nutritional value of grass-fed beef resides in the fat fraction. Bone broth made from collagen, rich bones through extended simmering, knuckle joints, femur with marrow, chicken feet releases glycine, proline, and hydroxyproline into